KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
NEET, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.in.
Good morning students. This video I would like to explain about muscle contraction. Previous video we discussed about muscular system introduction, especially skeletal muscle. When we are showing anything movement, how muscle is contract and relaxation. When you show that hand movement, our bicep will be extend and relaxation, extend and relaxation. How it will contract and relaxation. So how that mechanism is happens. That mechanism of the muscle today I will say in the muscle contraction explanation. Okay, already in the previously muscle containing with two different filaments, thin and thick filaments. Thin filament is a actin filament, thick filament is a myosin filaments. These concept once revise it. Okay, then only you will understand about the muscular contraction concept. Okay, let me start today video session. See the mechanism of muscle contraction. What is this thing? In the introduction time already I said when we are performing several movements, muscle want to contract and muscle want to relax. Okay, that only facilitate the movement and locomotion of our organism. Okay, of organism. These total muscle contraction explained by the one theory. That theory is sliding filament theory. Several theories are there. Especially in our syllabus, they are given the sliding filament theory. This is an acceptable theory for different people. Okay, so sliding filament theory proposing by the Huxley and Hansen, Huxley and John Hansen. These two scientists are studied about the muscle movement, how the internal arrangement of the muscle, that internal structures, how it will be contract and relaxed of muscle contraction, they are explained. So especially a statement they are given at the time of the muscle contraction, sliding of the thin filament over in the between the filaments, especially thin and thick filaments are overlapping each other. Okay, thin filaments we know and thick filaments also we know, correct now. So these two filaments are overlapping. That overlapping only to leads to the sliding filament theory. So the total sliding filament theory, especially in the muscle contraction, explained by the 5 steps of the muscle contraction. That 5 steps are first step excitation of the muscle, second step formation of the cross bridges, and then power stroke, recovery stroke, and relaxation of muscle. These are the 5 steps. When the muscle contraction is started and then get it to the relaxation time, these 5 steps are sequence order it will be happen. Okay, first step what it is, excitation of the muscle, second step formation of the cross bridges, third step power stroke, fourth one recovery stroke, and then next relaxation of muscles, fifth one relaxation of muscles. What it will happen and each and every step, finally the combination integration of the all these different 5 steps leads to the muscle contraction. So first step we want to know, in the first step excitation of muscle. Excitation of muscle. What will happen? So here for example somebody's brought tea. On the time you want to take tea with your hand. That is a action. Now that action is controlling by your hand movement. I mean controlling by means done by your hand movement. Now when you start to move of your hand, first process excitation of muscle want to occurs. Okay, now that excitation of muscle step, what happened mean muscle contraction initiated by the signals are sent by the central nervous system. Okay, when you seen that tea glass or anyone, okay, anything, your brain is, your eye will be observe that image, that information is sending to the your brain. Your brain is giving to the one order, take it that a tea glass, tea tumbler, like. So your brain, our brain, especially brain is initiate the one signal, that to central nervous system onwards a signal will be initiated. Now that initiated the initiated signal from the central nervous system onwards, it is carrying to the appropriate organ. Appropriate organ mean here for example if you want to take a tea tumbler with your hand will get the information, should not, our leg should not get any information. If you want to move from little bit distance means first legs will be getting information. So basing on the situationally, that activities causing signals are initiated by the central nervous system. That central nervous system onwards appropriate organ, that message, that signal is transported by the motor neuron. In 10th standard you studied neurons are 3 types: sensory neurons, motor neurons and mixed neurons. Which of the neurons are taking the information from the receptors to brain? Those neurons we call as a sensory neurons. That means when you are take out tea tumbler, before that thing you seen that tea tumbler, correct na? That tea tumbler image we get it in the brain. On that time when the visual information is going to up to brain by the sensory neurons. By analysis of our brain is giving the order, go and get it, that tea tumbler. On that time from the brain onwards your hand and your legs want to take information. So on that time that signals from the central nervous system, appropriate organ, messages are carrying by the motor neuron. Now got it. So through motor neurons, signals are carrying by these motor neurons and reach that appropriate organ. Especially here we are discussing about the muscle kadanana. So now we are taking that muscle. Okay, so the signals are reaches neuromuscular junction. Neuromuscular junction mean neuron end position synaptic cleft is there, correct na? So which is the organ want to show activity, that organ having the muscle. So neuron and that muscle junctional places, a junction point between the neuro and muscle tissues that we call as a neuromuscular junction. So totally what it is carrying the information, that total information is giving to the muscles. Up to now information here neurons, correct na? Now that neurons onward signals want to give it the muscles, but these muscle and neurons does not contact directly. There is no any direct contaction. In between the neuron and then muscle a small gap is there. That gap only we kept the name neuromuscular junction. Now the information passing time, here neurons are releasing that information by the one chemical substances, that is a neurotransmitter. That neurotransmitter best example acetylcholine. For example, here if you need some clarification mean how electricity and then how cell phone towers are working, like that. Electricity wise we need electric supply of something, wires connections are necessary, but cell phone signals wise no need wire connections, wire avasarame ille. Okay, in the through the wind also the signals will be pass out, correct na? From a signal cell phone tower onwards, our phone having the something connection through the wind contact, by that signals will be reached. Like that here, neuromuscular junction time, neuron releasing the something signals, that too in the which form, in the chemical form, that chemicals we call the neurotransmitters. That neurotransmitter example acetylcholine. Now that acetylcholine to reach the muscle will be polarized, muscle will be activation. Okay, nothing but coco play time, one person sat on that line, another person will give the then it will be start to activate, then that person will be move on, correct now? Like that neuron is giving the signals to muscle in the form of the neurotransmitters. Okay, so generate the action potential. Then on the sarcolemma, action potential generated on which place in the sarcolemma? You know one statement, which of the plasma membrane located or surrounded the muscles, that muscle sarcolemma, that muscle plasma membrane we call as a sarcolemma. Sarco is a nickname of the muscle. So on the muscle sarcolemma action potential are present. Now that sarcolemma onwards, that action potential want to spread out the total muscles. So here that action potential spread out through the inside of the muscle with the triad system. Triad system. Remember this statement, Nana. This word in the next video you will get what is the triad system. Three things mingle to each other to form the triad system. Okay, that three things, one is T tubes and cisternae. T tubes and cisternae. Cisternae is nothing but endoplasmic reticulum. In the previous video I said sarcomeres and muscles are containing with endoplasmic reticulum. That endoplasmic reticulum we call as a sarcoendoplasmic reticulum, correct na? So these triad system onwards, action potential to send out or spread out the all the muscle. Okay, that only here I mentioned. Through the T tubes, these action potential will spread out. When the action potential spread out the total muscle fibers, SER, sarcoendoplasmic reticulum, cisternae are releasing the one ion. That ion is calcium 2+. Okay, calcium 2+. This is the first step, excitation of muscle. Okay, somebody has said something information, good news, automatically you will get something excitation, correct now? Then like like that here at the end of the excitation of the muscle, calcium ions are released in which place? Inside of the muscle. Muscle structure theriyum kada nanna. Muscle fascicles ulla muscle fiber ki ulla myofibrils. Now here inside of sarcolemma is present on the around the muscle fibers. So that muscle fibers contains these, getting the calcium ions. That is the first one. So in the first step what you have to remember, by the central nervous system initiated signals to reach the neuromuscular junctions. That neuromuscular junctions are releasing the neurotransmitter, that is the acetylcholine. Then it will be spread out through the triad system, through the total triad system. By the end of the excitation of muscles, calcium ions are released. Then now come to the next point. What it is released the calcium ions, those things continue of the muscle contraction. Here formation of the cross bridge. Cross bridge means simply I will tell only one thing. That thing is actin and myosin to touch each other to form the just connections. Okay, attaching to each other. When the actin and myosins are attaching to each other, this is the time to recall. Myosin is a hockey-like structure, correct na? Actin is a something thread-like structure. Now both are having the binding sites, correct na? The hockey-like head of the myosin will attaching with the actin. Next video, I will draw the diagrams, Nana. That will give something more clarity to you. So, see that crossbreed formation of the crossbreeds. In the crossbreed formation time, initiated by in the first step, what it is releasing the calcium ions, that calcium ion concentration increasing in the muscle. Calcium ion concentration increasing in the muscle. So, when the very more volume of the calcium ions are increasing in the muscle, that a calcium ion bind with the troponin. Troponin is one of the protein, subunit of protein in the F filament, correct na? F actin filament. In the regular interval places, F actin filaments containing the troponin. That troponin having 3 subunits, TNT, TNC and TNL, correct na? TNI. So, these TNC is a place for binding of these calcium ion. By the binding of these calcium ions, important activity is started in the thin filament, that actin filament. By resulting of the binding of the troponin C bind with the calcium ion, troponin and tropomyosin complex move away from the active sites of actin. Active sites of actin, up to that active sites of actin is covered by the tropomyosin. Because of the round structure, actin filaments, globulin filament covered with tropomyosin. Okay. So, when that troponin binded by the calcium ion, that initiated change the something structural arrangement of these actin, troponin, tropomyosin structures. So, by that troponin, tropomyosin complex move away from the active site of the actin, then active site will be exposed. Active sites are exposing means it is willing to bind with myosin. So, that is a chance in the myosin filament head bind with ATP. Here when the actin myosin want to bind means they need something energy. So, energy wise myosin filament head bind. Head hockey stick like head myosin having the 2 sides, actin binding sites and ATP binding site. Especially in that ATP binding site, ATP will bind. When the ATP is bind, myosin getting energy. So, by using of that energy, myosin head bind with the active site of actin. And to form the cross bridge. So, how it is means just when you see any bridges observation time, some of the bridges build up in time, they make the several pieces. Okay, bridge is a several assembling of the several pieces. So each and every piece edges places they are keeping the something gap. Okay, that is a reason for some physical science reason is there. When that summer season extension will be there roads, so that's why they are keeping something gap. Now, what is the concept mean? When you see in that gaps, it is having the something attachment of the bits of that bridges. Like that, this is also actin is one bridge piece and then myosin also one bridge piece. Both are joined together, but not permanently, not detaching permanently. So, they are very close to form the one bridge. Like that, that too it is a cross manner. So, that is why we given the name is a cross bridges. So, here by the attachment these already ATP is present in the myosin. By the ATP binding only myosin getting the energy. Kada? So, now when the cross bridge formation after that ATP is started to hydrolyzation. Hydrolysis means breakdown of ATP. Breakdown of ATP or ATP reacting with the water molecule. So, when it is binding with water molecule, start with reacting with water molecule, ATP is split into the ADP and phosphate. Here ATP means adenosine triphosphate, correct na? 3 phosphates are there. ADP means adenosine diphosphate, 2 phosphates. So missing of one phosphate, that missed one phosphate in the form of the inorganic phosphate. Inorganic phosphate. That is a second step. Increasing in the second step, formation of cross bridges. That cross bridge formation time initiated by the calcium ions. That calcium ions are releasing by the cisternae of the sarcoendoplasmic reticulum. That calcium ions are binding with the troponin, that is troponin C subunit. By that resulting of these binding, then troponin tropomyosin complex will move away from the active sites of actin. That is a chance to myosin will bind with the actin. For binding efficiency, myosin first bind with the ATP. That ATP binding after the myosin head will be getting energy. That will be binding with the actin. So that binding with the actin time, it is act as a formation of appearance as a cross bridges. The final end of that these step, hydrolysis of the ATP. ATP will split it into the ADP and inorganic phosphate. Then come to the next point. Next step, power stroke. Power stroke anna forcibly muscle proteins want to dislocate. I mean dislocate means their position onwards they want to move somewhat of forward. That time power stroke want to necessary. Okay, nothing but when hockey person, he is touching the ball, hockey ball, but he is not at kicking anything ball. Simply he is roaming with bat, with the help of the bat. Okay, they are moving with the ball with the help of the bat. But when he is want to goal, he want to use the power stroke. Okay, he want to lift the hockey bag somewhat of back and then you want to kick that ball. Correct now. Like that cross bridge is formed. Now the cross bridge formation after the muscle want to contract na. So contraction time muscle proteins want to move the somewhat of forward. So that's why cross bridge is pulls inside, attaching the actin filaments. Okay, so like already I explained the example way how hockey player is moving the ball to throw the like that attachment of the actin filament towards of center of the A band, towards of A band. Okay, that A band, when you see in the A band, when you remember that A band, middle of the A band, what it is there Terimanana, H zone is there, and some places darkness is there, and actin filaments or myosin filaments are overcoming that some places. By that A band, now that A band moving the both edges onwards, sarcomere therimanana, off light band, both sides and full A band kada. That either sides onwards of I bands will become closerly. That means Z line will come closer. Here both sides of Z lines come and closer. So in the between of the A band, in the inside of the A band, H zone is there now. That H zone will be disappear. That only here I mentioned. Thereby the causing of the shortening of the sarcomere contraction. So when the power stroke happen, cross bridges are pulls that actin filaments in between of the myosin filament. Actin filaments goes inside of the myosin filaments. So that's why finally it leads to the shortening of the sarcomere. Now very important statement, I band size will reduce. That means half portion of the I bands of both sides in the sarcomere, correct now? That both sides onwards I band half parts will be enter into or towards of then nothing but moving into the in between of the myosin filaments. So I band reduce in the size, A band not yet reduce in their size. So in the muscle contraction, A band never change their size, length. Okay, oftenly I band reducing and getting normal position, reducing their size, getting normal position, that will be occurs. In the A band, in the A band, H zone will be disappear. These two statements you should be remembered, Nanna. One is towards of center of A band, both sides on Z lines will come closer. That is the first statement. By the closing of the Z lines, sarcomere will contract, shortening. At the time of the shortening, I band reduced in their size. I band reduced in their size. A band will not change. At the time of changing of reducing of the I band size, H zone will be disappear. That is a thing. Okay. And then end of the power stroke. At the end of the power stroke, ADP will release. ADP will release. Now again that myosin is free with bind with some other ATPs. Okay. Then next one, recovery stroke. In the recovery stroke, the myosin get back to their original position. That means one more example here. When you see that Onam festival time in Kerala, some of the people boat festivals are conducting. At the time when the boat want to move forward, the people will be using a oar pull the water, correct now? When the water pull and then they want to take that oar into the back side. So this is myosin also will come to the back side, that we called as a recovery stroke. Okay, the myosin get back to their original position, it is a relaxed stage. Okay, then finally when it is a recovery stroke, cross bridges will broken. Cross bridges is broken. Okay, so joint between the actin and myosin filaments are breakdown. Then last one, relaxation of muscle. Okay, right. Now you totally tea is taken. Okay, tea glass is over. I mean, tea glass is empty. Now your tea tumbler want to kept in the back side. Up, you are stretch your hand. Correct now. When you stretch your hand, relaxation of muscle will occur. When the motor impulse are stopped. Okay, from the brain onwards there is no symbols, so signals. Lack of signals, it is stop to generation of calcium ion. Here if signals are there, means calcium ions will generate. So here when the impulse are stopped, these calcium ions are pumping is stopped. Instead of the pumping, they want to collect the calcium ions. Up to now calcium ions are in the muscle, that too attaching with the myosin, troponin subunit, correct now. So what are the calcium ions are binding with the troponin subunit? Those calcium ions are everything came back. Okay, they are reverse way. Okay, then that uptaking and pumping back into the sarcoplasmic reticulum cisternae. Here cisternae onwards releasing now. The same cisternae is getting the calcium ions. Okay, lack of calcium ion, muscle contraction will not occur. This is the muscle contraction mechanism. Okay, first one, excitation of muscle and formation of cross bridges, power stroke, recovery stroke and relaxation of muscle. Okay, that means when you want to start of one activity for movement of any one body organ, okay, that first central nervous system signal is giving to the appropriate organ, then muscles will be to show that these 5 steps movements. Finally muscle contraction will occur. We are also, by the muscle contraction, we are also complete our activities in the general life. This is the muscle contraction. Okay, right. Next, diagrammatic explanation of muscle contraction I will explain. Okay, and along with this we will send one video. Okay, one animated video. By watching of that video, you will get some more clarity regarding mechanism of muscle contraction, because of it is a visual understanding is more helpful to you for this one. Okay, right. Thank you, Nana.